Chapter 3, Section 8, Principles of Respiration, the Electron Carriers. Now remember, respiration is the taking of the electrons, moving electrons everywhere. It's based on this whole idea that you're going to take electrons, strip them off of one molecule, transfer them to another, in the process maintaining that high energy state of the electrons. Redox reactions. Somebody gets reduced because somebody got oxidized. Oxidized means you lost the electron. Reduced means you gained the electron. This idea of electron carriers, though, you can't have high energy electrons, electrons in a high energy state just free floating around the cell. They'll have a tendency to bind to, glom onto any stray molecule they come into contact with. Inorganic phosphate, proteins, amino acids, nucleic acids, all sorts of weird things. No, you need to have some form of what they refer to as an electron carrier. You see electron carrier, think chaperone. Okay, the chaperones of choice are NAD and FAD. They will take the electrons that are harvested in glycolysis and citric acid cycle, and they will transport them somewhere. If it's a eukaryotic cell, they'll transport them to the mitochondria. If it's a prokaryotic cell, they'll transport them to the cytoplasmic membrane. Yeah, even bacteria do some form of oxidative phosphorylation with the cytochrome complexes and the ATP synthase. NADH dehydrogenases and flavoproteins. These are going to be electron transport proteins in the membranes. They're going to utilize this idea of oxidation reduction, taking the excited electron and passing it along. In the process, bleeding off a little bit of that energy. You've heard, like cytochrome C and whatnot. That's one of them. There's going to be dehydrogenases, flavoproteins, iron sulfur proteins in these protein complexes. Remember, anytime you hear the phrase protein complex, that means a collection of proteins, each having their own roles that have to be together in one giant unit. Individually, or that unit, that complex, minus one or two of the proteins, has no function, will not work. You have the dehydrogenases, the flavoproteins, the cytochromes that are directly interacting with those excited electrons, bleeding off the energy, and then you're going to have proteins that are known as quinones. They do not interact directly with the electron carriers or with any of those that are transporting, moving the electron and bleeding off the energy. They're more for maintaining structure, transport of, things like that. This dehydrogenase interacts directly with NADH. Its job is to take that excited electron off of NAD, to oxidize NADH, and then start it into this process of the electron transport chain. Flavoproteins do the same thing, but they interact directly with FAD. Our flavin mononucleotides, which is a new one, its job is to accept right there and right there two hydrogens, 1, 2. You take this ribitol and you turn it into adenine monophosphate, AMP. You have our friend FAD, which serves a role just like NAD does. We've talked about that in previous sections. FAD and here FMN can handle 1, 2 electrons. Remember, yes, they're showing hydrogens, but it's an excited electron pulling along a proton. The cytochromes are nothing more than proteins that have a heme, prosthetic group, a heme. That means they can take the electron, bleed off the energy, and then transfer it along. Remember, heme is an iron center. It takes and then passes it along. And here's what it looks like. Heme B. Okay, this is going to be enclosed in a much larger collection of polypeptides, a much longer protein. Here's the iron that's going to take that excited electron. When it takes the excited electron, it's going to cause everything around it to change its conformation, which will then allow for the transport of a hydrogen across the membrane. Others organisms are going to use, can use iron, they can use sulfur, Fe2S2, Fe4S4, and whatnot. They can also use ferredoxin. Okay. There are multiple mechanisms for this bleeding for proteins and their little helpers, for lack of a better term. There are multiple ways this can be done because there are multiple organisms out there that have alternate methods other than just the one that you've learned in cell or intracell and molecular. Remember, not everybody likes oxygen. So not everybody's going to use the same cytochrome complexes like we do because they have no use for oxygen as a final electron acceptor. So they're going to use alternate methods. Maybe they're also going to use alternate cytochrome complexes or ferredoxin complexes or flavin proteins or something else. I already talked the cytochromes, so I'm not going to go into those too much.